Welcome back to class. I hope everyone had a little refresher, whatever that means for you, on Wednesday. We're going to continue with the nervous system today, picking up where we left off on Monday. And today is going to be our last day covering general background of the nervous system. On Monday, we're going to move in and talk about the brain specifically. Before we get into it, one last reminder. I'm hiring students and looking for students to do research for credit and or pay. This spring and summer. If you're interested or know someone who is, please pass along the word. There's information in Handshake about that. I'm going to take this down next week. So if you're interested, act now. Okay, today's agenda. First thing I want to do is debrief your lab practical. You probably saw your grades were emailed to you, I think, through. Did it automatically email you? Okay, I was trying something new. And then the grades are posted in Blackboard. And then I'm going to start off by reviewing Monday's Ticket Out, just a couple concepts to get your brain thinking about your brain. And then I've got a human anatomy in the news. So at the end, or not the end, but in the middle of last class, you asked about microglia and where new microglia come from. This led me down this whole rabbit hole, and I found some really cool research about that, and I want to share that with the class. And then we'll continue talking about neuronal integration. So basically the ways that signals are information is brought together in different parts of the nervous system. And then I have yet another human anatomy in the news. So weird day today. We have two human anatomies in the news. It's Friday. It's good. And then I've got actually I don't have a ticket out today. I should change that. But I will wrap up with some announcements and reminders about our schedule. I've got a Venn diagram. And then Dr. Vadney is going to come for the last few minutes of class and plug her neuroanatomy class with you a little bit. So she's going to join us at the very end. So busy agenda today. Don't forget, you've got a study guide on the nervous system. It's the Google Slides file that you can modify as you like. Before we dive into all of this stuff, what questions do you have on this Friday morning? Cool. All right, well, let's do it. Lab practical number 1, you saw your grades. As I mentioned, the lab practicals are in my opinion, and this is borne out by student grades, the most challenging aspect of this course. Here you can see the distribution of grades across. So we had a handful of students, 3 total get a grade above a 96 or above. So that's pretty sweet. Otherwise, you see a pretty even distribution here. I mean, there's kind of a cluster in the 70s and 80s. So that's how things panned out for the class. The class average was just below an 80, about 78%, which is actually quite a bit higher, a fair bit higher than previous classes, the average from previous classes. So overall, the class did well on this. The median was 82, so that's the middle grade about here. And you can see the range there. So I'm going to present this information so you kind of get a feel for where you fall. I know at the lab practical, it could be a little bit challenging because you can't really go back and check your answer because you don't have the thing in front of you anymore. If you have specific questions, though, I can go over that with you, and you can do that in open hours or during lab next week. So let me know because I know that you might want to revisit that. Know that our second lab practical is only covering information on labs from here forward. So it's not cumulative. It's just the first and second half of the class. So you will not be assessed on this material again. The exception being that certain anatomical terms I want you to know and bring forward in class. So the regions of the body, terms like proximal, distal, all of that kind of stuff. Of course, we'll be revisiting those, but you won't have specific questions on the muscles of the upper limb or anything like that. Questions on the lab practical? Okay. On Monday, on your way out, I asked you some questions, and I want to revisit these questions and make sure we have good understanding. One of the questions was, which of the following cells produce myelin? Now, remember, myelin is this insulative lipoprotein that wraps around the axons of neurons to insulate them and allow them to conduct their impulses efficiently and quickly. And the class by and large got this right. 100% of you said oligodendrocytes, which are the myelin-producing cells in the central nervous system. And most of you said Schwann cells, which are the myelin-producing cells in the peripheral nervous system. So remember on Monday, we talked about those glial cells. These are nervous system cells, but not neurons, so they're not transmitting information. We talked about 4 types of glial cells in the central nervous system and 2 types in the peripheral nervous system. And remember that these are the two types that are producing that myelin in those two systems. So good job there. My next question for you, it was most sensory neurons are, now sadly, none of you picked polar bear, but whatever. Most of you got this correct. So the answer here is unipolar. And so just to refresh your memory, because most of you got this correct, but only about 2/3 of the class, I want to bring you back to this slide. Now, remember that generally these sensory neurons, with some exceptions in the special senses, are unipolar or pseudo-unipolar, they're sometimes called. And so in this case, they have this cell body that kind of just branches off the axon there, right? So just to refresh your memory there, that most of these sensory neurons are unipolar. Now, the second follow-up question was, which is true of a sensory neuron? And here, again, about 2/3 of the class got it right. And that is that it has a cell body in the peripheral nervous system and terminal boutons in the central nervous system. So remember, a sensory neuron is delivering information from generally outside the body to the CNS. So those terminal boutons where neurotransmitters are released is going to be in the CNS. And if we go back to this diagram, you can see kind of schematic example here where we have the peripheral nervous system, we have the end of the neuron where information is being received. We have the cell body branching off, but still in the peripheral nervous system, probably in some sort of ganglia or ganglion singular. And then the axon continues into the central nervous system, and those terminal boutons are in the CNS. So a couple of takeaways to get here is make sure you know the structure of a neuron, a sensory neuron that is unipolar, and make sure you understand which part of that neuron is in the CNS versus the CNS. Questions on that? All right. Well, I want to talk about this microglia thing for a little bit. And I want to focus on one study in particular that came out last year, I think it actually was. And here you see, this is Lee Gann, who is the lead researcher of this group. And her group did this really cool study with mice. And I have a figure here from the paper that I don't really understand. The paper had a whole bunch of really complicated figures, but I just figured I'd include a figure. That's poor language. I thought I'd include a figure here. Don't worry about the details here. The takeaway message is they did this experiment with mice. Now, we can't do these experiments in humans for obvious ethical reasons. So a lot of the work that's done understanding neurophysiology and neuroanatomy is on mice. And actually, Dr. Vadney, that's going to come visit our class later on today, does a lot of work in that area. And the idea being that if it's true in mice, we generally assume that it's more or less the same in humans. So we did this cool experiment with mice. Now, microglia, remember, are those immune-derived cells in the central nervous system. And what's interesting is that excessive microglia, a condition known as microgliosis, is associated with a whole bunch of neurodegenerative diseases. In other words, it's thought that because there's something going on where your neurons are degrading, that these microglia are being activated, right? And so if you see a lot of microglia in your brain, too many of them, it's probably a bad sign. The question is then, where do these microglia come from? And there's basically three possibilities that these researchers tested. They tested whether other glial cells can start translating certain proteins and essentially become these microglia, or if they are derived from other immune cells that are formed in the bone marrow and then brought to the brain via the circulatory system. So they are created in the bone marrow and then they circulate in the blood up to the brain. Or do existing microglia in the brain replicate and form these new microglia? So what they did is they used a method, then they wiped out 90% of the microglia in the mouse brain. And they found in a relatively short period of time, they actually came back. So they reduced their number of microglia down to 10%, and boom, soon enough, the mouse had a full array of microglia. And it's not worth getting into the experimental details, but basically what they did is they added fluorescent tags to different cell types, and they could actually track where they move in the body and the brain, which is super cool. But the takeaway message here is they found overwhelming evidence that the microglia that repopulated the brain came from those existing 10% of microglia. So in other words, mature microglia are replicating and producing more microglia when they need to. So by conjecture, it's thought then that like this microgliosis, this abundance of microglial cells associated with neurodegenerative disease, is the result of those mature adult, those mature microglial cells regenerating themselves. So cool study, and I was excited to find this directly relevant to a question that came from class. So note that there's a link to both the popular press article and the original research article in your program. You don't have to read the original research article. I put it in there for your reference. I do recommend reading the popular press article about this. It will take you all of 4.5 minutes, if not less. And it's pretty interesting stuff. And you can expect a question or two on quizzes and exams about your human anatomy and news. Cool. I'm glad this question came up. This was neat. And I didn't know the answer, but I was excited to find some new and exciting research that asked exactly that question. Questions, thoughts, comments? Cool. Neurons. Let's get back to them. So very important distinction here. Often in our common everyday speech, we conflate the words neuron and nerve. Anatomically speaking, they are different things. They are related, but they're very different. And so it's important that we watch our language here. Nerves are cable-like organs in the peripheral nervous system formed by the axons. So a nerve is essentially a bundle of axons of neurons in the peripheral nervous system. So we commonly talk about nerves because on a gross anatomical scale, that's what we see and that's what we kind of interact with. For example, if you whack your funny bone on your elbow, you get that tingling sensation because you've whacked your ulnar nerve there, right? But that nerve is a structure that's composed of a whole bunch of axons bundled together. And the structure looks like this in cross-section. You can see a diagram here. You can see that there's individual axons, and of course you can see the myelin sheath around those axons. They're bundled together, or excuse me, each of those individual axons is wrapped in a layer of connective tissue called endoneurium. Then this bundle of axons is wrapped together in something called perineurium that surrounds what's called a fascicle of axons. That term probably sounds a little familiar to you. You have blood vessels, and then around the whole nerve is this epineurium. So we have individual axons wrapped in endoneurium, then those groups of axons in bundles called fascicles that are held together by something called perineurium, and then the entire thing is kept together by this outer layer of connective tissue called epineurium. If this terminology sounds familiar, I hope it does. That's because it's exactly the same terminology we saw in muscles. We have individual muscle fibers being bound by this endomysium, and we have these fascicles bound by perimysium, and we have the whole muscle tissue being bound by epimysium. So those same terms, endo, peri, and epa, for the muscles are exactly what we see here for a nerve, just to make some connections there. We will look at cross sections of nerves in lab and microscope slide, and you can see these individual bundles, and you can see how they're arranged. It's pretty cool. And that's all we really want to say about the anatomy of those nerves for now. Can expect a diagram. Almost every year, I think I include this diagram or some variation of it on a quiz or exam. So be sure to know that. What I want to do now is take a step up anatomically at a larger scale and talk about two different kinds of nervous tissue that are commonly called white and gray matter. You've probably heard this terminology before. White and gray matter are the names of the two primary kinds of nervous tissue in the central nervous system. And you can see a diagram here. This is a cross-section of your spinal cord, right? You can see, let's see, what is this? This is the terminal bouton of a sensory neuron coming in here in the back. So we have an interneuron, and then we have a motor neuron bringing signals out. And you can see in this cross section very clearly this kind of almost butterfly shape of darker matter surrounded by this lighter matter. And that is representative of the color you'll actually see when we look at some cross sections of spinal cords. And so what I want to talk about is what those different kinds of matter are composed of. So gray matter that's found in the center of the spinal cord. And I'll talk about gray and white matter in the brain when we get to the brain next week. For right now, I want to just define these two terms and show you examples of where they are in the spinal cord. Now, the gray matter is a mixture of cell bodies and dendrites, and you can see that in the diagram here, as well as these short non-myelinated interneurons. So we talked about the importance of myelin for helping signals travel long distances. In some cases, in many cases, these short neurons called interneurons that are found entirely within the central nervous system, they're super short, so they don't need this myelin. So they're non-myelinated neurons, right? We also find the synapses in this gray matter because that's where those neurons are communicating with each other. Notably, this gray matter, we don't really see anything here that's myelinated, and that's why it's gray. So the simple reason why the white matter is white is because it contains the axons of myelinated neurons, and that myelin is white in color, and so that makes this white matter white. We don't find that in gray matter, and so it's darker in color. In the central nervous system, gray matter is mostly on the inside of the tissue. So a good example we see in cross-section of the spinal cord here. The exception is that in the cerebrum and cerebellum of the brain, we find a layer of gray matter on the outside as well. Your cerebral cortex is a gray matter. And like I said, we'll talk about that in more detail when we get to the brain, but just know for now that in general, this gray matter is found deep in the central nervous system, and the white matter is found more superficially. So let me pause there on this slide, and then I want to talk in a little more detail about what the white matter is made of. Okay, white matter does not contain cell bodies, but instead just has all of these axons, most of which are myelinated, hence the white color, that run along different parts of the central nervous system. So there are these tracks of myelinated axons. Note that a lot of these are myelinated, but not necessarily all of them, but still there's enough that are myelinated that it makes it look white in appearance like this. You can see on the diagram here, this white matter is the superficial matter of the spinal cord. This white matter also contains lots of neuroglia. So these are those support cells. So for example, what kind of neuroglial cell do we expect in association with all of this myelin around these axons? Oligodendrocytes, good. Yeah, what else do I want to say? Within the central nervous system, we have these bunches of axons running together, kind of from one location to another in the same, more or less the same place. We call those tracks or commissures. So these are similar in a sense to nerves in the peripheral nervous system, which are these bundles of axons of peripheral axons of neurons in the peripheral nervous system. In the central nervous system, they're not in this bundle formation like they are in the peripheral nervous system, but they're found kind of in these groups. And if they run kind of up and down the body on the same side, they're called tracts. And if they switching and they're crossing over from left to right, they're called commissuries. And we'll see details of this when we get to the brain again. But for now, I just want you to know that those are these groups of myelinated axons that run together in this white matter. Cool. So that's the basic, the kind of gross anatomical structures. I want to talk for a minute about how these things work together in something called neuronal integration. And again, this is getting into a bit more of the physiology than anatomy, but what I want you to take away from this is those major gross anatomical structures of the nervous system that are involved with neuronal integration. Because after all, neuronal integration is the whole point of the nervous system, right? And so anatomically, we can easily define the central from the peripheral nervous system. But I want to note here that functionally, they really can't act one without the other, right? We can say, okay, the central nervous system is the brain and spinal cord, and peripheral nervous system is everything outside of that. But what's important is that they're constantly working together. They're not two independent systems doing their own thing, but they're essentially one would be useless without the other. And just as a way of a bit of review, we have these afferent fibers of the peripheral nervous system carrying input, sensory information to the central nervous system. So you see that here. Oh, this is bad slide design here because I put that red. I should have put the word afferent in blue to match the color of that nerve there, but I didn't. But afferent is sensory, remember? And then we have these interneurons that are these generally short neurons found entirely inside the central nervous system. And they often integrate this information. It's often a very complex network of these interneurons, but I'm just showing you a simple one here. And then that transmits information to the efferent fibers of the peripheral nervous system, which produces some kind of response. And generally, we're just going to talk about a motor response, but of course that can be good. So information goes in, it's integrated in the central nervous system, goes out. This is all review. We kind of started the whole central nervous system unit talking about this, right? What I want to do now is take this and get in a little more detail and see how this looks functionally. And specifically, since we're not going to talk about the brain just yet, I want to talk about a reflex arc, which uses all of these aspects of this system, but doesn't involve the brain at all. So by definition, a reflex arc is the simplest form of neuronal integration, and it's responsible for your reflexes, right? And these are just these very quick autonomic motor responses. They can be visceral or somatic, but they are things that your body does before you even realize you do it. And in many cases, that information doesn't even get to the brain until after the action is completed. So there's some pretty simple examples of this. One is the reflex when you touch something hot. When you touch, actually, I just did this yesterday. I touched the hot pan accidentally with my fingers, and your hand will pull away from that before you realize that you're being burned, before that information even gets to your brain. And it's not until after your hand is removed that you realize why you jerked your hand away. Another example is we have a reflex that if we hear a loud sound, we turn our head in that direction. And so if you hear a bang off to your right, you'll turn your head and look that direction, generally speaking, before you even realize that you've moved your head. You won't realize you've done it till afterwards. Of course, it all happens within a fraction of a second. It's very quick, but these are reflexes, in this case, that are utilizing this sensory input. Now, we also have, well, let me give one more example, but let me talk about the reflex arc here. So there's 5 parts of a reflex arc you see here. The first is the receptor, where that information is originally received. So in this case, we're looking at a receptor associated with the skin. Then we have this sensory neuron that delivers that information to the central nervous system. Notice it's a sensory neuron, so it's unipolar. You can see its cell body right here. In this case, bless you, we have a whole bunch of these sensory neurons, and these cell bodies are going to be grouped up here in something called the dorsal root ganglion in this case, and we'll see examples of that in the lab. That sensory neuron then brings that information to an interneuron within the gray matter of the central nervous system. So notice here that the axon, the long axon of the sensory neuron, is myelinated. Out here in the peripheral nervous system, it's part of a nerve, and then it goes through, it's part of this white fiber of the central nervous system, and then terminates in, the white matter of the central nervous system, and then terminates in the gray matter, where it drops some neurotransmitters that are picked up by this interneuron, found entirely in the gray matter, where that information is integrated. And then that in turn sends the signal out this motor neuron, step 4, and that reaches an effector organ, in this case, some kind of skeletal muscle. So there's 5 parts to that. And you can see the legend here for the circle, and these represent the cell body. And then we have the synapse here is represent these terminal boutons that are this kind of Y-shaped thing. So you can see that the sensory neuron has its cell body outside the central nervous system. The terminal boutons are here. The motor neuron, however, has its cell body right there in the gray matter of the central nervous system. So that different structure between sensory and motor neurons you can see in this diagram as well. Now, the simplest form of a reflex is called monosynaptic, and that is a reflex arc that actually doesn't even have this interneuron. So it skips this whole step, and information kind of amazingly travels directly from the sensory neuron to the motor neuron. And because it has only one synapse, because it has less parts, it's the fastest kind of reflex. So an example of this that you're familiar with is the knee-jerk reflex. So this is something you probably have had your doctor do. We'll do this in lab, actually. I have the little mallet, so we don't even need one. You can just use your hand. And what happens here is when you're sitting and your knees, your legs are relaxed. I'm going to sit on this table and hope it's strong enough. That tendon and those ligaments that go from your patella down your tibia here are kind of loose. And if you take something and you tap them, it's hard to do it to yourself, kind of until you do it. What it does is it tightens those ligaments, and you have a reflex that contracts the muscles of your quadriceps and jerks your knee up, right? It's usually not like this, it's not dramatic. It's usually just like a little movement, right? The reason for that is because we talked about the nervous system delivering information from where our body is in space, and how much the ligaments and tendons are stretched and muscles are stretched, so we know where our limbs are. What's really curious about this is a good example of if this tendons are rapidly contracted when you're walking, it's because you're probably tripping or falling. So it's actually kind of a tripping reflex that your body thinks that you're tripping, and so your leg kicks out to stop you from falling. Now, obviously, in this case, you're sitting, you're not actually falling, but it's that same reflex arc that produces that movement, again, before you even realize it. So go ahead, Evan.
Someone with like cerebral palsy and they lack like fine and gross motor skills,
Mhm.
do they still have this reflex occur? Or not?
That's a good question. I don't know the answer. Let me think about it for a second. My guess is that yes, because primarily we're in that case, you're affecting the integration center in the brain itself. And in this case, the signal never goes to the brain, but I would have to check that. That's just a guess. Yeah, that's a really interesting question. Yeah. And that's why actually, so on that note, that's why the doctor will check this reflex, because if it doesn't work, that indicates that something is going on between the peripheral and central nervous system here, and that signal is not being transmitted. You know, as far as CP goes, I'm not sure exactly. Good question. So that's a monosynaptic reflex. Again, it's simple and it's fast. We also have what's called a polysynaptic reflex, which is much more common. And this is the example I gave earlier, this withdrawal reflex. And this is called polysynaptic because here we have this interneuron. So we have two synapses, synapse between the sensory neuron and the interneuron, and a synapse between the interneuron and the motor neuron. And this, I gave the example of touching something hot. This can also be touching something sharp or other kind of intense pain stimuli. And that's why we feel pain. We feel pain to protect ourselves. In a simple physical sense. I'm not talking about emotional pain. That's different. I have to take a psychology class for that. In terms of physical pain, the reason that things hurt is because they're going to damage our body, and then we avoid them. If we touched hot things and it didn't hurt, but it totally destroyed the tissue, and let's say our hand, if we touched it, that'd be bad news for our survival, because we'd be doing dumb stuff all the time and we wouldn't even know it. We'd end up injuring ourselves. And there's a disease for that, and that is part of the symptoms of leprosy. So leprosy is the disease where often people like lose parts of their extremities or have damage to that. A lot of the reason for that is because they lose their pain receptors in their fingers or their extremities. And so essentially you do things that are damaging your tissue, but you don't know it, so you keep doing them. So it sounds, as much as being in pain is not fun, the alternative kind of sounds a lot worse, to continually damage your body but not even know it until it accumulates pretty seriously. So that's a polysynaptic reflex. There's lots and lots of examples of that. We can talk about that a little more in lab, but for now, just kind of know the anatomical structure of that in those 5 pieces of a reflex arc. I have one last slide on neuronal integration, and I want to go back to our little interneuron here. We see this very simple path of information in this case, where we have this one interneuron between the sensory input and the motor output. Well, most of the time, outside of these reflex arcs, we have much, much more complicated ways of integrating information. And I'm just going to touch on this super lightly at the moment, for the moment. But you can see different arrangements of interneurons here. We have these different circuit paths. So we have what's called a diverging circuit, where we have an input that's spread amongst many, many interneurons, and each interneuron spreads it to more than one interneuron. So it kind of forms this network of neurons that are transmitting the same information. We have a converging circuit where we have a whole bunch of neurons delivering different information to the same neuron. And then we have what's called a reverberating circuit, where you have this kind of cyclical motion of information, where information is being transmitted to other neurons, but also then transmitted back. And so I'm not even going to begin to touch on the complexities of the way this information is transmitted through the brain. The takeaway message here is that these configurations of interneurons can be very, very complex and lead to very complex paths of information through your brain. And this is important because we're constantly integrating information from our senses of sight and smell and hearing and all of these things on a super fast scale that we don't even think about, so that we can navigate through our world. And that happens because of these complex networks of neurons. And just to give one example of this, I want to return to this pain reflex. What's important to note here is I talked earlier about just this reflex arc, this withdrawal. It's curious here, now they have like a match system instead of a tack. It'd be fun to make these textbooks and just think about, like, you can just come up with like, all right, what pain sensation can the person in the diagram experience next? And so I talked about this reflex arc here. What's important to note is that information, in addition to traveling through this reflex arc, also is transmitted through neurons in the central nervous system up to the brain. And so this is why you know that your finger touched something painful and then was withdrawn, because the information doesn't just stay in that reflex arc, it also gets transmitted up to the brain and integrated. And so that's why a split second later, you kind of look around, you realize what's going on, and then you can respond to it. The information goes up to the brain, in this case, the sensory cortex. I mentioned that there's gray matter on the superficial aspect of the brain. You can see that here. And then that signal in turn is sent back down the spinal cord and interacts with that motor neuron to get your arm to do something in response to getting burned, right? In this case, you're going to be like, oh shit, I just burned myself. Find some cold water and run it on your hand. And be like, that was dumb. I'm never lighting a match and watching it burn down on my finger again. Hopefully, depends how well your integration center in your brain's working. So the takeaway message here is we have this reflex arc, and information is traveling through the central nervous system and back down to allow us to have these more long-term responses beyond just that initial reflex. So we have that initial reflex to get us out of harm's way, and our brain is integrating all of this information to help us figure out what to do in the longer sense. Yeah, Kelly.
So when people who are diabetic and like lose like those feelings, does that just happen like at like the receptors? So when they like step on something like hot or like they don't realize it, is it like damage at the initial receptors or is it like damage somewhere else on it?
Where does that pathway get damaged? So you're talking about leprosy, right? The example I gave earlier where you lose that sensation.
I just know
Yeah.
that people with diabetes in my family has a history of diabetes.
Oh yeah, diabetes can affect it too. Yeah, so how does that work? It's a little bit different in diabetes than leprosy. Let me get back to you on that one. Let me look that up and give you a better information or a better answer in Monday, because that's a really good question. And I think I have an idea, but let me confirm that. I'll make a note for myself. So thinking about these pathways and thinking about the example I gave earlier in this week, we watched that short video, actually last Friday it was, about the TED Talk where you can control the arm through this, because all of this information is in electrical circuits. I want to give you a couple more examples here of some recent advances that have allowed people where this system isn't working properly to integrate information into their central nervous system. So I've got a couple, actually 3 examples here of this. So a new study allows electrical sensors in an artificial limb to transmit information to the brain. So think about having an artificial limb. Think about having an artificial foot. Even if that foot was stable, it was lightweight, it was comfortable, a huge issue is that you're not getting any information. When we walk, we're constantly getting information from the bottoms of our feet and our legs to tell us what kind of ground we're moving on, if it's stable or unstable. We're constantly adjusting our stride and our gait to compensate. But an artificial limb, you don't have that, no matter how good that limb is. Researchers have been able to integrate electric sensors in the foot and transmit that information to the central nervous system. So I put a link to this popular press article in your program for Monday. Hey, Dr. Vadney, how you doing? Good, come on down. I'm just gonna finish up a couple of slides here and then we'll talk about neuroanatomy. A second study actually does the opposite. Researchers have figured out a way to put these implants, so this is a model, but they put implants associated with the person's brain, and that allows that person to, instead of, in the case of the prosthetic, which is sending sensory information back to the central nervous system, in this case, they're providing a system whereby someone's thoughts can be translated into these electrical signals and then allow them to operate something that allows them to move. So this is an example of a French guy who was in a car accident and became quadriplegic. And now with this technology, I'm going to show you this video, he can actually use this suit to allow him to walk. And it's a pretty cool example. I want you to check this out. I'm going to finish this up and I have a perfect transition to talk about neuroanatomy. So, you know, obviously you can see it's a big clumsy suit. It's tethered to the ceiling. It's not something that this person is going to use on a day-to-day basis just yet. This is a first step in allowing this to happen. But he's walking. He is controlling that suit with his mind, is what I really want you to take away from it. It's not the case that the suit is moving his body. Well, he's controlling the suit and then the suit is moving his limbs, right? But otherwise, that information wouldn't be transmitted to his limbs. So it's kind of a workaround and allowing, again, those electrical signals of the central nervous system, in this case, to communicate directly with the computer. And it's pretty remarkable if you think about, you know, this person being able to essentially walk across this room by this interface of his brain with the computer and then this computer controlled suit. And so I put an article about that in your program as well. And I just want to throw one more at you as well. And this is, as this technology improves, we can do it at a finer and finer scale. And in a similar program, they were able to implant the same kind of idea in a monkey and allow the monkey to control individual fingers. And so this is important because we think about those fine motor skills where those individual muscles and individual motor units are being activated. It's easy to make a whole bunch of muscles contract together, right? We saw that in the video when they send the electrical signal down that ulnar nerve and the whole hand just kind of goes like this. This is an important advance because it's allowing this monkey to move individual fingers through this kind of artificial circuit. So it's another good example. So I put links to those 3 popular press articles in the program for Monday. Check them out. It's interesting kind of applications of the stuff we're learning here. And that is a perfect transition. I'm going to skip the brain. We'll get to this. Let me just give you a couple quick reminders, and then I want to transition to Dr. Vadney here. On Monday, we have a quiz in class. Don't forget, it's closed book, 20 minutes. We'll do it first thing. It covers the skull and everything we did in the central nervous system through Monday this week. So it won't include stuff we talked about today. And then your lecture exam is the following Monday. It'll be online, and we won't have a regularly scheduled class. Details are in your class program. Danny's got tutor sessions, and I've got open hours. I've got to leave a little bit early on Monday, but I'll be there from noon to 1:30 on open hours. Now it is Friday, so I have a Venn diagram to share. So this circle here, actually, let me pause. I don't want to get there yet. I want to see what questions you have before I wrap things up here. We're good. Course schedule makes sense. Quiz for Monday. You ready to rock it? Sweet. All right. So this circle represents everything there is to know about neuroanatomy. This is your Venn diagram for today. This circle, it represents what you learn in this class in human anatomy. Notice that there's very, very little overlap here, meaning that we cover very, very little neuroanatomy in this class. But neuroanatomy is super cool. And so if you take Dr. Vadney's neuroanatomy class, you're going to learn a whole bunch more neuroanatomy. Now, I didn't put this circle the same size because I figured you're not going to teach everything there is to know about neuroanatomy, but it's pretty close. And I want to just point out that there's very little overlap. So we touch on neuroanatomy just a little tiny bit in this class, but if it's something you're interested in, which you should be interested in, then
Yeah, so thank you for letting me crash your human anatomy course. So I'm Chelsea Vadney. I'm a faculty member in psychology and neuroscience. And I have been teaching for a little while now this course, neuroanatomy. And so if you found neuroanatomy interesting in this section that you covered in Dr. Gangloff's class, I would encourage you to think about taking neuroanatomy. If you've thought, man, I really want to understand more about the structures in your nervous system that are allowing you to have all of these complex functions that make you human, I would encourage you to think about neuroanatomy. We're talking about structures that are important for regulating emotion, homeostasis, learning and memory, movement, sensation. So we start out by kind of covering gross neuroanatomy, so those major structures that you can see by the naked eye, and then we talk about different parts of the brain in more detail and circuits that allow you to have various functions. And then if you are interested in studying anatomy because you would like to review it by holding an actual human brain, last year and hopefully this year we will be able to continue to rent human brains from OSU. And so there'll be an opportunity to do that as well as part of this course. So if you want to explore more about what is inside your heads, I would encourage you to think about taking neuroanatomy. I'm happy to answer any questions that you have about it. You can email me at cavadney@owu.edu. I have some handouts here that I didn't print a whole lot of them, but if it's something that you're interested in, my email's on the bottom of it. It's just basically the slide with my email. And my office is over in Phillips Hall in the psychology offices in 52D. I'm also happy to kind of talk about any questions you may have about neuroanatomy or neuroscience related courses or psychology courses. Do you have any questions for me about the course? Yeah. Yeah, so it's currently only offered in the spring, and actually next year I'm going on a sabbatical for my 4th year, so this would be a great time to take it, so it may not be offered next year.
I have a logistical question. I know that for like the zoology majors, they're required to take a certain number of upper level zoo courses. Do you know if this counts for one of those?
I don't believe it does at the moment,
No, it
yeah,
doesn't.
so it's an elective for neuroscience,
Right.
and that's pretty much what it counts for at the moment, but.
I think there, if someone was in this situation, I think it, I mean, it should count for an upper level bio course. And I think there's a good case to make a petition. So I would be great to throw in support anyone that take this class and like, courses. And that's a better example of an upper level biology course. So if that's an issue, I would be happy to talk more about that. Support.
Thank you. Any questions for me?
Do you know what time you'll be teaching this class? Next week?
Yes, it'll be from 1:10 to
Oh, I see.
2:30 on Tuesdays and Thursdays.
Sorry.
No, you're fine. Yeah, the time is different this year from what it was the past 2 years. I'm also teaching molecular and cellular neuroscience, so we just kind of swapped the times for that to better accommodate the neuroscience majors.
Cool.
Well, thank you for having me.
So no ticket out today, guys, no nothing, just go study for your quiz on Monday. There is a class program with the readings that I have here and some other details about upcoming course schedule. Have a great weekend. Halloween weekend too.